For all things health and fitness, this is the Army Gymnastics Podcast.
The Army Gymnastics Podcast.
Discover a fitter, healthier you at armygymnastics.com.
Welcome to the Deep Dive brought to you by Army Gymnastics. You're here because you're on that journey, right? Towards personal excellence, always pushing to understand more, to be better. Today, we're taking a really deep dive into a topic that, well, it affects billions of people globally, often silently, but it carries these really profound implications for our health, our well-being, and honestly, our very understanding of human biology. We're talking about herpes reactivation and viral warfare, the hidden science. Now, I know, for many of you, just hearing the word herpes might bring up thoughts of frustrating, often hidden challenges, things that can subtly undermine our goals or quality of life. But here's the powerful truth we really want to get across today. While these battles are often waged invisibly, down at the cellular level, science is constantly shedding light on them, peeling back the layers of mystery to reveal this incredibly intricate, almost like viral warfare happening right inside our own cells. Our mission today is to empower you with that knowledge, to strip away some of the mystery and show you just how understanding this hidden science could truly be a game changer, not just for how we approach herpes, but potentially for a whole range of other complex diseases that impact all of us.
That's exactly right. And in this deep dive, we're going to unpack some truly groundbreaking discoveries, stuff from just the past year or so. We're not just focusing narrowly on herpes itself, but really on a much broader, more sophisticated understanding of how viruses, generally speaking, are, well, far more cunning manipulators of our genetic machinery than we ever really thought before. We'll explore the really unexpected ways these viruses reactivate. We'll look at the molecular battlegrounds inside our cells where this viral warfare actually unfolds. And maybe most importantly, we'll discuss the surprising new strategies that are emerging in the fight against these incredibly pervasive pathogens. This deep dive, it's designed to offer you a real shortcut, a way to become genuinely well-informed on a topic that is, quite frankly, shaping the very future of medicine and how we think about health.
Okay, let's unpack this. It sounds fascinating. Our starting point, this ubiquitous intruder, is the herpes simplex virus, or HSV. And when we talk about how common this virus is, the numbers are, well, they're genuinely astonishing. I mean, just think about this for a second. Over 60% of people under the age of 50 worldwide are infected with HSV-1. 60%.
Staggering.
That's the one mainly known for cold sores, right? But it's also increasingly causing genital herpes, too. We're talking about over 3.8 billion people. Billion, with a B. And then there's HSV-2, primarily linked to genital herpes. Over 10% of people globally are infected with that one. These aren't just abstract numbers. These are billions of lives touched by this virus. It's just so pervasive, it's almost hard to wrap your head around the scale.
It really is. And it's crucial to understand, like you said, that HSV-1 and HSV-2 aren't just, isolated oddities. They belong to this much larger family, the herpes viruses.
Yeah.
You might be surprised who else is in this family.
Yeah, like who?
Well, the varicella zoster virus. That's the one that gives us chickenpox when we're kids, and then can come back later in life as shingles.
Right.
Also, Epstein-Barr virus, EBV, famous for causing mono or glandular fever, and cytomegalovirus, CMV, another really common one that usually doesn't cause problems unless your immune system is weakened.
Wow, okay, so they're all related, and they all share that key feature, right? The ability to stick around for life.
Exactly, that's the common critical threat.
Yeah.
They establish lifelong infections. Once they get in, they basically never leave. What's absolutely fascinating from a biological standpoint is how HSV manages this lifelong persistence. You see, once the virus enters your body, it never truly goes away. Our immune system is incredible. It does a fantastic job of controlling it most of the time. It sends the virus into hiding, into what we call a slumbering or latent state.
So it's asleep, basically.
Kind of. During this latent period, the person infected often has no symptoms at all. No cold sores, no blisters, nothing visible. But the virus isn't gone. It's just lying low, out of sight, dormant.
And where does it hide?
It actually takes refuge inside our own nerve cells, which is incredibly clever, really. For HSV-1, the cold sore type, it typically hides out in nerve cells in the trigeminal ganglion. That's a nerve cluster near your ear, with nerves branching out to the face.
Oh, okay.
And for HSV-2, the genital herpes type, it usually settles into peripheral neurons, nerve cells down in the lower spinal cord area. These nerve cells become a permanent reservoir for the virus, just sitting there, waiting for a chance to reactivate and cause an outbreak.
And we know some of the triggers for that reactivation, right? Like stress.
Yeah, absolutely. Stress is a big one, physical or emotional stress.
Mm.
Other infections, too, if they temporarily weaken your immune system. Even things like intense sun exposure, getting a sunburn, can trigger it for some people. Those are the sort of classic triggers we associate with a flare-up, the times the virus decides to wake up.
And the consequences. Most people think of cold sores or genital sores, which are definitely annoying and uncomfortable. But it can be more serious than that, can it?
It certainly can. HSV-1, for example, in rare cases, can cause viral encephalitis. That's a severe inflammation of the brain. It's very dangerous, potentially life-threatening, and can cause permanent neurological damage.
Wow.
And here's something really thought-provoking that's emerged more strongly in recent years. There's growing, quite compelling evidence linking HSV-1 infection to the development of Alzheimer's disease later in life.
Alzheimer's. Really? From the cold sore virus?
The link is being seriously investigated. It's not definitive proof of causation yet, but the association is there, and it's strong enough that researchers are looking very closely at it. It really shifts how we think about the long-term systemic impact of these seemingly simple viruses. Could they be playing a role in neurodegeneration? It's a huge question.
That changes everything. Okay, so traditionally, how have we dealt with herpes? Just managing symptoms.
Pretty much. The mainstays have been antiviral medications, drugs like acyclovir, villisclovir, famciclovir, and they are quite effective. They manage symptoms well, they reduce the frequency, the severity, and the duration of outbreaks. They work by inhibiting viral replication, basically stopping the virus from making copies of itself when it's active.
But they don't actually get rid of it, right? They don't cure it.
Exactly. That's the critical point. They don't eliminate the virus from its hiding spots in the nerve cells. They don't prevent it from staying there, latent, waiting for the next chance to reactivate. So it's effective symptom management, but it's definitely not eradication.
Which brings us to something really crucial. You mentioned earlier the sort of sneaky nature of reactivation. It happens more often than we think.
Far more often. And here's the real kicker. It frequently occurs without any visible symptoms at all.
No sores, no blisters, nothing.
Nothing you'd notice. This is called asymptomatic viral shedding. The virus becomes active, starts replicating and shedding from the skin or mucous membranes, but the person feels completely fine.
Wow, so you could be spreading it and have absolutely no idea.
Precisely. And this silent shedding is a major reason why HSV is so widespread. It explains how it transmits so easily, contributing to those incredibly high prevalence numbers you mentioned. Studies have really shown a light on this. Some research showed that across all participants, viral shedding happened on average about 18% of the days they were tested.
Nearly 1 in 5 days.
Yeah. And even in people who said they were completely asymptomatic when they joined the study, shedding was detected on 10% of days.
So that old idea of just being either active with sores or latent and silent, it's not really that simple, is it?
Not at all.
Yeah.
We're moving towards what some call a noisier view of HSV infection. The virus is likely active at a microscopic level much more frequently than we ever realized, even if we don't see or feel anything. And this constant, often unnoticeable activity means the virus is continually interacting with our cells. And that, as we're about to see, has even more profound implications than we thought.
Okay, this is already fascinating. But now you mentioned groundbreaking recent discoveries, things that changed the picture even more.
Absolutely. If you thought the prevalence and the asymptomatic shedding were mind-bending, wait until you hear about how HSV actually manipulates our cells. We're finding it's not just a passive intruder hanging out. It's acting more like an opportunistic interior designer for our cells. It's actively re-engineering things.
An interior designer. Okay, I'm intrigued. What does that mean?
Let's start with a really surprising discovery from the University of Virginia Anna Cliff's team. This was published just February 2025. They found something astonishing about how HSV reactivates. You might think the virus just waits for, say, your immune system to dip because you're stressed, and then it jumps out.
Yeah, it makes sense.
But their research suggests it's much more active than that. Get this. The virus deliberately makes a specific protein called UL12.5, and this protein actually triggers our own body's immune response in the nerve cell where it's hiding.
Hold on. It triggers the immune response. Why would it do that? That sounds like poking a sleeping bear, like setting off the alarm yourself when you're trying to hide.
Exactly. It seems completely counterintuitive, doesn't it? You'd think activating the body's antiviral defenses would be the last thing the virus wants to do. It should signal danger, invite an attack.
Right.
But here's the incredibly cutting part. The virus subverts that very defense mechanism it triggers. UL12.5 is the first viral protein ever identified that is absolutely required for HSV to wake up from dormancy. And it does this by manipulating the antiviral process within the infected neuron. Instead of the immune response hurting the virus somehow hijacks that response to create the perfect conditions for its own reactivation.
That is. Wow. So it turns our own defense system into its wake-up call.
Essentially, yes. It's an amazing example of viral manipulation, and the implications are huge for treatment, because now we have a specific viral protein, UL12.5, that we can target. The idea is if we can block this protein, maybe we can prevent the virus from waking up in the first place.
And targeting a viral protein would hopefully mean fewer side effects for us compared to targeting one of our own proteins.
That's the hope, a much more targeted strike. And importantly, they found that HSV2 also makes this protein, so this discovery could apply to both major types of herpes simplex. It really shows the virus isn't just passively waiting, it's actively sensing things, making this protein and taking control of our cellular responses for its own benefit. It's disturbingly smart.
Disturbingly smart is right. Okay, so that's one level of manipulation, but you also mentioned it physically messes with our DNA, like redecorating the place.
Yes, and this is where that interior designer analogy really hits home. There's another stunning study, this one from Pia Cosma's team at the Center for Genomic Regulation, published in June 2025. They discovered that HSV1 literally rearranges the 3D architecture of the human genome inside our cells, and it does this incredibly quickly within hours of infection.
Hours. It rearranges our DNA structure.
The structure, yes. How our DNA is folded and organized in the nucleus. And this isn't just some random side effect of the infection. It appears to be a deliberate strategic action by the virus. Its goal: to gain rapid and easy access to the specific host genes that it needs to replicate itself.
So like you said, an unwelcome guest rearranging your whole house to build their own workshop, moving walls, pulling out wiring.
Exactly, that's a great analogy. And this study started to figure out the molecular tools the virus uses for this renovation. Within the first hour, just 60 minutes after infection, HSV1 performs this incredibly audacious move. It hijacks a key enzyme called RNA polymerase II, or RNAP2.
RNAP2, isn't that essential for reading our own genes?
Absolutely essential. Think of it as our cell's master copy machine. It reads our DNA blueprints and makes RNA copies, the instructions our cells use to build everything they need. But HSV1 basically commandeers this vital machine and forces it to start making copies of viral genes instead to synthesize the virus's own proteins.
Wow. Okay, so it steals the copy machine. What else?
It also recruits other key players. Another enzyme called topoisomerase I, or TOP1. Normally, TOP1 helps manage our DNA by snipping and resealing it to relieve twists and tangles, keeping it organized.
Like untangling a rope.
Exactly. But the virus ropes TOP1 into its project too. And even cohesin, that's a crucial protein complex that acts like molecular glue, holding loops of our DNA together, really important for organizing the genome cohesin, also gets pulled away from our DNA towards the viral genome. All these hijacked host factors, RNAP2, TOP1, cohesin, are then used by the virus to build these structures called viral replication compartments, or VRCs.
Viral factories, basically.
You can absolutely think of them like that. Highly efficient viral factories, assembled right inside our cell's nucleus, right where our own DNA is supposed to be in charge.
And what happens to our own DNA while this hostile takeover is happening?
The consequences are pretty dramatic. It's not just a minor disruption. It's like a wholesale theft and a complete architectural collapse for our genome. Because RNAP2 and TOP1 are busy working for the virus, they effectively abandon our human DNA. Transcription, that vital process of making RNA from our own genes, just collapses across the entire host genome.
So our own cell's instructions basically get shut down.
Largely, yes. And as a result of this transcription collapse, our human chromatin, the DNA packaged with proteins, gets dramatically compacted. It gets crushed, as the researchers describe it, into this incredibly dense shell. It shrinks down to only about 30% of its original volume, and it gets physically pushed to the very edges of the nucleus. Out of the way, essentially.
Making it hard for our cell to access its own genes.
Exactly. And here's a really surprising twist that came out of this research. For a long time, the dogma was that dense, compacted chromatin automatically means genes are shut down, inaccessible. But this study suggested it might be the other way around in this case. It's the act of stopping transcription first that actually causes the DNA to compact so dramatically.
So the activity, or lack of it, drives the structure change, not just the structure dictating the activity.
Precisely. It reveals this complex, dynamic two-way street between gene activity and genome structure that we're really only just beginning to fully appreciate. The virus isn't just passively using our stuff. It's actively and aggressively remodeling our entire nuclear environment to maximize its own replication while shutting down our defenses.
That is just incredible manipulation. But you mentioned finding a weak spot. Did this study offer any hope on that front?
It absolutely did. And this is potentially huge. They found something crucial. If you block just one of those hijacked host enzymes, topoisomerase I, the TOP1 enzyme that untangles DNA, it completely stops HSV1 from being able to rearrange the human genome.
Just blocking TOP1 stops the whole redecorating process.
It appears so. Without TOP1 function, those viral replication compartments, the viral factories, they just fail to form properly. And the result in their cell culture experiments was striking. The virus couldn't produce a single new infectious particle. Zero.
Wow. One enzyme holds the key.
It seems to be a critical linchpin in this process. And this makes TOP1 a really, really attractive new therapeutic target.
Yeah, I can see that. If you can stop the virus from setting up its factories.
Exactly. It could lead to entirely new types of antiviral drugs, maybe not just for herpes, but potentially for other viruses that might use similar genome manipulation strategies. It's a move beyond just managing symptoms towards actually disrupting the virus's fundamental ability to replicate.
This is shifting my whole perspective on viruses. Okay, so HSV is a master manipulator. Does this kind of viral warfare on our genome happen with other viruses too?
Oh, absolutely. This isn't unique to herpes, although herpes viruses seem particularly adept at it. Let's broaden the view a bit, drawing on insights from a big review paper looking at the dynamics of viral and host 3D genome structure during infection. It starts with understanding that our own DNA, in eukaryotic cells like ours, isn't just a long string. It's incredibly organized in 3D space within the nucleus. You have these large domains called TAIDs, topologically associated domains, where DNA tends to interact more within itself, and you have specific loops forming, like enhancer-promoter loops, where distant regulatory elements come together physically to control gene activity.
So the physical shape and folding of our DNA is critical for how our genes work.
Exactly. It's a fundamental level of gene regulation. And what this review and many other studies now show is that viruses across the board have evolved ways to exploit and actively modulate this host chromatin architecture. They do it to suit their own life cycle needs, maybe to help them establish latency and hide, or maybe to ramp up gene expression during the lytic or replicating phase. They're literally messing with our cellular blueprints layout.
And are there common targets, like things they all go after?
Yes, certain architectural proteins seem to be frequent targets. Two key ones are CTCF and cohesin. CTCF acts kind of like an insulator, setting up boundaries between different genomic regions, and cohesin, as we mentioned, is that molecular glue essential for folding DNA and holding loops together.
The same things HSV was manipulating.
Precisely. Because these proteins are so fundamental to our own genome structure, they become prime targets for viruses wanting to rewire the system for their own benefit.
Okay, so give us some more examples. How do other viruses wage this chromatin warfare? Let's stick with the herpes virus family first, the HHVs.
Sure. Their large DNA genomes often form those circular episomes we talked about, like mini chromosomes inside our nucleus. And these episomes actively recruit our host cell's regulatory machinery. Take Epstein-Barr virus, EBV, the monovatic, is a classic example. It can control its own genes by changing its episome structure. For instance, it can silence one of its key genes, EBNA2, by forming a specific loop within its own viral DNA.
So it can turn its own genes on and off using our machinery. Clever.
Very. But it also messes with our genes. A big one is the MYC proto-oncogene. MYC drives cell growth, and when it's overactive, it can lead to cancer. EBV can actually boost MYC expression in our B cells.
How does it do that?
It seems to weaken the normal chromatin looping that keeps MYC in check, essentially letting it run wild. This involves interactions between the virus's own enhancers and our MYC gene, and EBV episomes don't just sit there, they physically interact with our host chromosomes. When the virus reactivates, its episome can actually move from silent, repressed parts of our genome, heterochromatin, to active, open regions, euchromatin, to get better access to the machinery it needs.
That's incredible. What about other HHVs, like KSHV, the one linked to Kaposi's sarcoma?
Similar story with KSHV. CTCF and cohesin binding on its episome can help keep the virus latent by restricting lytic gene expression. But, like EBV, it's also implicated in increasing MYC expression, contributing to the cancers it causes. And for HSV1, which we discussed, and also cytomegalovirus, CMV, CTCF binding seems to play roles both as an insulator and in promoting lytic transcription, depending on the context.
So across the herpes virus family, it's a recurring theme. Manipulating our genome architecture proteins, like CTCF cohesin, to control their own genes, using their viral products to mess with our gene enhancers promoters, and physically interacting with our chromosomes.
That's a great summary of the key HHV strategies. It's a multi-layered attack on our genome's organization.
Okay, what about viruses outside the herpes family? You mentioned HPV, human papillomavirus, and its link to cancer. Does it do similar things?
Yes, but with a twist. HPV genomes can exist as episomes, like herpes viruses, but they can also integrate directly into our host chromosomes. And that integration event is often a key step towards cancer.
How does integration lead to cancer?
Well, normally an HPV protein called E2 helps keep the viral oncogenes, E6 and E7, under control. These are the genes that drive cell proliferation in cancer. But when HPV integrates, the E2 gene can get disrupted. Or sometimes the integration disrupts CTCF binding sites that normally regulate E6, E7. Either way, you can lose that negative regulation, leading to overexpression of E6 and E7, which pushes the cell towards becoming cancerous.
So the integration itself messes things up.
It does. But it's even more insidious than that. Integrated HPV DNA can cause major structural rearrangements in the host chromosome. It can actually hijack host enhancers. Imagine an enhancer that normally controls a tumor suppressor gene, one that stops cancer. The integration event can physically rewire the DNA looping so that this enhancer is now driving a pro-proliferative gene instead, like CCDC106, helping to fuel tumor growth.
Wow, so it's not just inserting itself, it's fundamentally reprogramming the local genetic landscape to promote cancer. That's deeply manipulative.
It really highlights how changes in 3D genome architecture triggered by the virus can be a direct mechanism driving oncogenesis.
Okay, those are all DNA viruses. What about RNA viruses? Do they get involved in this chromatin warfare too, even if they don't usually integrate their genes into ours?
They absolutely do, which is perhaps even more surprising. Let's take HIV-1 first. Now, HIV is an RNA virus, but it's a retrovirus, meaning it does convert its RNA genome into DNA and integrate it into our host genome. So it's a bit of a special case among RNA viruses.
Right. Does where it integrates matter?
Very much so. HIV doesn't just integrate randomly. It shows a strong preference for integrating into active open regions of our chromatin, often near clusters of superenhancers. It picks prime genomic real estate.
So it wants to be where the action is, transcriptionally speaking.
Exactly. And the level of expression of the integrated virus, the provirus, is heavily influenced by the epigenetic state and the 3D organization of the chromatin where it landed.
Okay, so HIV integrates strategically. But what about RNA viruses that don't integrate, like, say, the flu virus?
Influenza virus, IAV, is a great example. It doesn't integrate its RNA genome into our DNA, yet it still manages to significantly alter our host 3D genome architecture. One way it does this is through its NS1 protein. NS1 can cause something called read-through transcription, essentially making our RNA polymerase II ignore normal stop signals. This runaway transcription can physically disrupt chromatin loops and even kick cohesin off CTCF sites.
Making the DNA more open, more accessible.
Potentially, yes, making
Yeah.
certain loci more permissive to transcription factors that the virus wants to use. Another flu protein, NP, can interfere with a host protein complex involving cohesin, leading to the formation of abnormal chromatin loops that actually seem to enhance viral replication.
So even without integrating, it's finding ways to mess with the physical structure of our DNA folding.
Precisely. It's manipulating the machinery that controls that fold.
I have to ask, what about SARS-CoV-2? The COVID virus does do this too. It's an RNA virus that doesn't integrate.
Yes. Research indicates that SARS-CoV-2 infection also significantly impacts the host epigenome and genome organization. It's been shown to alter chromosome organization in a large fraction, maybe up to 30%, of genomic regions. This includes weakening or switching between active and inactive chromatin compartments. And critically, this might affect the expression of antiviral genes, potentially suppressing our initial defenses.
Could this relate to why some people get so much sicker than others?
It's definitely being investigated as a factor in COVID-19 severity. For example, remember we talked about CTCF. CTCF binding at the enhancer for the IL-6 gene seems to promote IL-6 expression. IL-6 is a cytokine, an immune signaling molecule that was heavily implicated in the hyperinflammation, the cytokine storm, seen in severe COVID-19.
Right.
Well, researchers found a specific genetic variant, a tiny difference in DNA sequence, an SMP, that disrupts CTCF binding at this IL-6 enhancer. And people with this protective variant were shown to have a lower risk of developing severe COVID-19.
Wow, so a subtle change in genome architecture interaction directly impacts disease outcome.
Exactly. It's a powerful example of how these viral host interactions at the level of 3D genome structure can have real-world clinical consequences. The virus might also cause a loss of cohesion, further disrupting genome organization.
So, okay, let me try to wrap my head around this segment. What it all means is viruses aren't just simple pathogens causing sickness. They're like molecular engineers, or maybe saboteurs, actively messing with the very structure and operating system of our DNA on a massive scale.
That's a really good way to put it.
Mm.
They engage in sophisticated, large-scale re-engineering of our genetic blueprint.
And understanding this hidden science, this warfare at the DNA level, is critical because it explains why these diseases can be so persistent, so complex, and often so hard to fully defeat. It's not just about fighting the virus particles, it's about dealing with the changes they make inside our cells.
Precisely. It's a much deeper, more intricate battle than we perhaps appreciated even a decade ago.
Okay. So now that we understand the enemy's tactics better,
Mhm.
are we figuring out new ways to fight back? What about new treatments, especially for something like herpes?
Yes, absolutely. This deeper understanding, particularly of how viruses manipulate our cellular machinery and epigenetics, is paving the way for some really exciting new therapeutic strategies. Let's start with epigenetics.
Right, the on-off switches for genes. You said latency involves the virus hiding its DNA using epigenetic silencing?
Exactly. The virus uses our cell's own epigenetic machinery to wrap its DNA up tightly as heterochromatin, making it inactive and invisible to the immune system. But this isn't a one-time thing. It's a dynamic process. Establishing latency, maintaining it, and then reactivating all of these steps heavily rely on our cellular epigenetic processes. How chromatin is assembled, specific chemical modifications to histone proteins like methylation or acetylation, methylation of the DNA itself, and even regulatory RNAs. The virus is constantly interacting with these systems.
So it's using our own tools against us, basically.
Yes. And reactivation often involves overcoming these silencing marks. Stress response pathways, for example, involving enzymes like JNK, can trigger signaling cascades that lead to the removal of repressive marks, or the addition of activating marks on the viral DNA, allowing viral gene expression to start up again.
Which means, if we understand these epigenetic controls, maybe we can interfere with them, keep the virus permanently silenced, or make it harder to reactivate.
That's exactly the idea behind epigenetic-based therapies, and there's some really promising work here. One area is targeting enzymes called EZH1 and EZ2.
EZ1 and EZ2. What do they do normally?
They're part of a complex called PRC2, the polycomb repressive complex 2. Its main job is to add repressive marks to histones, essentially telling genes to shut down.
So they help keep genes off?
Right. Now, researchers have developed inhibitors for EZA12 compounds like GSK126 or GSK343, and studies have shown that these inhibitors can actually suppress the oncolytic or active infection of herpes viruses in cells. They limit the expression of the first viral genes that get turned on, the immediate early genes, and reduce the number of viral genomes that are actively being transcribed.
So they help keep the virus quiet.
Yes, but interestingly, they do more than that. These EZ12 inhibitors also seem to boost our own cellular antiviral defenses. They increase the formation of PML foci, those little structures in the nucleus that help shut down viruses, and they upregulate the production of interferons, key antiviral signaling molecules.
So it's like a double whammy, suppressing the virus and boosting our defenses.
It appears so, and this suggests these inhibitors could have broad antiviral applications, potentially working against CMV, adenovirus, maybe even Zika virus, not just HSV. It's a strategy that leverages manipulating our own cellular control systems.
That sounds really promising. Any other epigenetic angles?
Another interesting area involves a specific modification on messenger RNA called M6A, or N6-methyladenosine. This is the most common internal modification on mRNA in eukaryotes.
Okay, a tag on the RNA message itself, what does it do?
It affects many aspects of the mRNA's life: its splicing, stability, transport, and how efficiently it gets translated into protein. And it turns out that many viruses, including herpes viruses, seem to hijack this M6A system. The modification can favor the expression of viral genes, maybe make their mRNAs more stable or better translated, and potentially it might even help the virus evade immune recognition by masking its RNA.
So if we could block this M6A modification,
The idea is that inhibiting the enzymes that add M6A, for example, using a compound called 3-deazodenosine, could have broad antiviral effects. The research is still developing, but it's another promising avenue for therapies that might target a process the virus relies on, hopefully without being too toxic to our own cells.
epigenetic therapies, that's a whole new way of thinking about antivirals. But what about the ultimate goal, a cure? Is that even possible for something like herpes that hides in our nerve cells?
Well, this is where things get incredibly exciting, possibly even paradigm shifting. We need to talk about gene editing,
Gene editing.
like CRISPR.
Similar technology, but in this case using something called meganucleases, which are like highly specific molecular scissors. There was a huge breakthrough announcement from Keith Jerome's team at the Fred Hutchinson Cancer Center just in May 2024.
Okay, what did they do?
They developed an experimental gene therapy. They used harmless modified viruses, AAV vectors, think of them as tiny delivery trucks, to carry the instructions for these meganucleus scissors directly into nerve cells.
The very cells where herpes hides out.
Exactly. The AAVs deliver the payload, and the meganucleuses are designed to find the latent herpes virus DNA within those nerve cells, and then they cut it.
They actually cut the viral DNA. Yes. The goal is to snip away at the viral genes, either damaging them beyond repair or potentially even cutting the virus out entirely. It's like sending in a microscopic repair crew to remove the faulty wiring.
That sounds like science fiction. Did it work?
The results in their mouse models were absolutely stunning. In mice with orofacial herpes, like cold sores, the therapy removed 90% or more of the latent HSV DNA from the trigeminal ganglia.
90%.
And in mice with genital herpes, it was even higher, up to 97% removal of latent HSV DNA from the dorsal root ganglia.
That is unbelievable. So it actually got rid of most of the hidden virus.
It appears so. And importantly, it didn't just reduce the amount of latent DNA, it also significantly suppressed viral shedding, the amount of virus being released. This suggests it could dramatically reduce transmission too.
Which would be huge from a public health perspective.
Absolutely. Now, there were some mild side effects seen at the highest doses initially, but they refined the approach, using a single type of AAV delivery truck, lowering the dose slightly, and using a promoter to make sure the scissors only worked in neurons, and this improved the safety profile while still being highly effective.
So the researchers themselves used the term curative potential.
They did, which is a very strong statement in this field. It's still experimental, of course, needs much more testing before human trials.
Yeah.
But it represents a massive leap forward, a potential path toward actually curing herpes by eliminating the latent reservoir. It's incredibly hopeful.
Wow. Gene editing for a cure. That's amazing. What about vaccines? We have vaccines for chickenpox, which is related. Why has a herpes vaccine been so hard?
That's a great question, and it's been a long-standing challenge. A key reason is, as we've discussed, HSV's ability to evade the immune system so effectively and establish that hidden latency in nerve cells very quickly after infection. It makes it really hard for a traditional vaccine to teach the immune system to mount a strong enough, fast enough response to completely block the virus before it establishes that reservoir. Past human trials have unfortunately shown limited success, often working well in animals but failing to provide strong protection in people.
But is there new hope on the vaccine front too? Maybe with newer technologies?
Yes, there definitely is. The whole vaccine landscape is buzzing with activity right now, partly thanks to the success of mRNA technology elsewhere.
Ah, the COVID vaccine tech. Is that being used for herpes?
It is. Moderna, for instance, has an mRNA vaccine candidate called mRNA-1608. This one is actually designed as a therapeutic vaccine.
Meaning for people who already have herpes.
Exactly. The goal isn't to prevent initial infection, but to boost the immune response in people already infected with HSV-2, hopefully improving their symptoms and reducing how often they shed the virus, thus limiting spread. It's currently in phase 12 clinical trials, with results expected around mid-2025.
Okay, that's interesting. What about a preventative vaccine?
BioNTech, another major mRNA player, is working on that. Their candidate is BNT163. This one is designed to be preventative, to train the immune system in people not yet exposed, hopefully stopping the virus from taking hold in the first place. That's in phase 1 trials, expected to wrap up end of 2025.
So mRNA is bringing new possibilities. Any other approaches?
Definitely. There's a company called Rational Vaccines that has several candidates based on live attenuated viruses, weakened forms of the virus. They have RVX-201 as a treatment vaccine and preventative candidates for both HSV-2, RVX-2001, and HSV-1, RVX-1001. Their early data, particularly from animal models for the treatment vaccine, showed a significant reduction in symptomatic days compared to controls, which is encouraging.
So it really feels like we're attacking this from multiple angles now: epigenetics, gene editing, advanced vaccines.
Exactly. It's a multi-pronged approach. We're moving beyond just managing outbreaks towards genuine prevention strategies and, perhaps most excitingly, towards the real possibility of a cure through gene editing. It feels like we're finally starting to crack the code of these incredibly persistent viruses.
This has been incredibly illuminating. So let's try to bring it all together. What's the big takeaway for someone listening to this? What does all this hidden science really mean?
I think the biggest shift is moving beyond seeing viruses as just simple invaders causing temporary illness. What we've uncovered, especially recently, shows they are incredibly sophisticated biological entities. They are master manipulators of our deepest cellular machinery, right down to the physical structure of our DNA. They're waging this constant hidden viral warfare inside us, actively re-engineering our genetic landscape.
But the flip side is, by understanding these incredibly complex strategies, these hidden mechanisms, we're finally finding their vulnerabilities, their weak spots.
Precisely. With new tools, new insights from genomics, epigenetics, structural biology, we're beginning to map out their playbook. And once you understand the playbook, you can start to devise effective counter-strategies.
Finding the Achilles heel, like blocking TOP1 to stop the factory setup, or cutting out the latent DNA with gene editing.
Exactly. These aren't just incremental improvements, they represent potential paradigm shifts in how we treat viral diseases.
And that's the final thought I want to leave everyone with. This ongoing battle against viruses, often silent and fought at the cellular level, it's completely transforming how we understand health and disease.
The breakthroughs we talked about today, understanding the 3D genome manipulation, the potential of gene editing, the promise of epigenetic therapies, these aren't just about herpes. They open doors to entirely new ways of thinking about and potentially treating a huge range of complex human diseases. Think about the viral links being explored in neurodegenerative diseases like Alzheimer's. Maybe similar cellular manipulations are at play there. Think about cancers driven by viruses, where understanding how they remodel our DNA is key to finding better treatments.
So the lessons learned from fighting herpes could apply much more broadly.
Absolutely. The more we understand this intricate microscopic warfare happening inside us, the better equipped we become. Not just to defend ourselves reactively, but to proactively reshape our health future, turning this deep scientific knowledge into real, tangible solutions that improve human lives. It's a really exciting time in virology and medicine.
Truly fascinating stuff. Thank you so much for breaking that all down for us. And for all of you listening, for expert fitness advice and personalized plans to help you on your own journey to excellence, visit armygymnastics.com. That's your go-to resource to reach your goals. Start your journey today at armygymnastics.com.
Thanks for listening. For all things health and fitness, subscribe to our newsletter today. Find out more at armygymnastics.com.